So good morning. I just want to welcome everyone. I'm Allison Bashir, the Dean of the UC Davis School of Medicine. And I first want to thank Ana Lucia and the entire team for all of their work in putting this longer presentation. I want to welcome you to today's Team Research Forum. And I want to take a few minutes to highlight that this is the UC Davis Collaborative Research Forum. And together we are helping to transform our society and improve health. This is our 77th research forum that grew out of the strong partnership between UC Davis Office of Research and the School of Medicine, bringing together our research teams and experts from across all of the disciplines and all of the schools at UC Davis. As a neurologist and researcher, I'm really pleased to share the School of Medicine's research growth over the last 2 years. It's been remarkable across so many disciplines. So in fiscal year 21, the clinical trials in research in the School of Medicine grew at approximately 63% in 1 year. The clinical trial awards are now at 98 million. UC Davis research for the entire campus hit a record of 968 million in research awards, a $27 million increase from last year. The School of Medicine's research awards are also on track with a 33% growth year over year, expected to top for fiscal year 21 at 368 million. None of this happens without teamwork, interdisciplinary research, and really a cross-campus collaboration. These research team forums that you're attending today started in April of 2020, when Vice Chancellor Prasad Mahapatra and I sat there and said, you know, we need to share all the information about COVID. So we put together what we thought would be a just one and done town hall, and now we're at 77. The School of Medicine and the Office of Research also collaborated to provide pilot funding to grow interdisciplinary research to really address COVID research. I'm pleased to report that as of July 30, we have had 322 proposals submitted to, for possible funding. 122 were awarded, and that is $46 million of new funding up to all of UC Davis. 31 million of that was from the School of Medicine, but many other parts of our UC Davis community are involved in research, and almost all schools have presented and are funded in this fight against COVID. We are all here to share information. This collaborative research has resulted in 386 COVID-related publications since March of 2020. Together, we're creating a national reputation for research in COVID at UC Davis. And nothing says that national reputation than our speakers today. Dr. Richard Mitchellmore, Dr. Brad Pollack, and Dr. Bart Weimer are joining us today. Their work has been featured nationally. I'm proud to say that UC Davis is one of the leading institutions for research for COVID. The School of Medicine is also one of the leading institutions on training a diverse population of physicians. And in fact, one of our greatest strengths is marrying that diversity and commitment to community with our commitment for research. And that's how we're going to really move forward in fighting this virus. So today's topic couldn't be more timely given the rapidly changing environment from day to day. We're impacted more by how COVID is now impacting our lives for the long haul. It's my pleasure to introduce the first speaker, Dr. Richard Mitchellmore. Dr. Mitchellmore is the founding director of the Genome Center at UC Davis and a professor in the Department of Plant Sciences, Molecular and Cellular Biology, Medical Microbiology and Immunology at the UC Davis School of Medicine. His work served as the foundation of the rapid asymptomatic saliva testing that is at the heart of Healthy Davis Together success. Has been nationally recognized as a model for protecting health. His innovative multidisciplinary research utilizes a synthesis of molecular genetics and evolutionary approaches. His work and expertise spans basic research and into molecular basis of specificity in plant pathogens and interaction with translational plant genetics. Welcome, Dr. Mitchell. We're so glad to see you. And I remember those days back in August of 20, and on behalf of all of us at UC Davis, we are so appreciative of the amazing work that you've done during this really unprecedented time.
Great, thank you. Hopefully you can see this.
Yeah, it's perfect. That's excellent. Thank you so much.
Great. Well, thank you very much, Dean Brashear, for that generous introduction. No, but what I will first say, it is almost to the day a year since we got the funding, and then a couple of days later saying, yeah, well, we do want to test everybody coming up onto campus in 6 weeks' time. It's been a tremendous team effort. I mean, I had the original concept. I like to think of myself, I'm the architect, but it's no good if you don't have some really skilled craftsmen to put it together. And it's taken a tremendous amount of dedication from people all across campus with the testing kiosks and in the lab that's worked very, very hard. But today I'm really just going to give you a brief synopsis of the testing and as a lead in to our analysis of variants using rapid SNP genotyping. I said there's been input from many people, particularly Charlotte Acharya, who's been one of the people in the lab that's really led this effort. Our goal has been to provide inexpensive high throughput testing and then to monitor the positives for variants of concern. And this technology really came out of what has been routinely used for over a decade in the ag biotech sector. And it's been in close collaboration with LGC Biosciences Technologies. So we're a temporary pop-up lab. We're an extension of the student health lab on campus, though we're just going through the process of getting our own CLIA certification. It was a repurposing of the DNA technology core. The MD is Ralph Green, who's been incredibly understanding and helpful. And if you want to see more details of what happens to your saliva once it enters the testing center, there's a very good YouTube video put together by Communications. So many of you may have been through this. It's really impressive. We've got 38 spitting stations, or we had. We're actually upgrading this to 42 in the fall. One of the ongoing discussions right now is how we can scale up to run even more tests than we've currently ever run. It is much more efficient than at-home collection, and we've had pretty high compliance over the last year. So the workflow for the testing looks something like this. The saliva comes in 96 format in barcoded tubes. We heat inactivate the virus. Working with saliva is problematic, but this was solved by the idea of Lutz Frönicke, who added papain, aka meat tenderizer, which is a remarkably effective protease that gets rid of the viscosity problem. And then we have a semi-automated workflow that takes the caps off, aliquots in the papain, shakes it, incubates it. Then we need to heat kill the papain. And then once we've aliquoted into 384 well plates, we can just stack them into this fully automated qPCR machine called an IntelliCube. And that does everything for us. It sets up the reactions and then runs them. And then we export the data up into the cloud, where using an AI-informed caller, it tells us, it really allows us to do efficient and high throughput calling, which is monitored by CLSs. So each machine can, we have 2 of these machines. We can do 768 samples in 1.5 hours, which in theory will give you 6,000 samples in a 12-hour day. But as I say, we can stack things into the machine so it runs overnight unsupervised. Now, what the situation looks like today, this is just the data off the Healthy Davis website. Most of the testing at the moment is coming through Healthy Davis together rather than the campus. The numbers are down there. But you can see that the number of daily positives, right now, we have more positives per day than we've ever seen before. The 70s have been our record. It was 47 before. And then if you look at the positivity rate, it's approaching what we've seen. So due to the Delta variant, it is causing a pandemic on a scale that is as high as we've ever seen. We've run close to 700,000 tests for campus, HDT, and others. Majority of the results are going back either the same day or next day. And the maximum number of tests that we've run in any one day has been just over 5,000, which is pretty impressive, but we are preparing, and I've just been in discussions this morning, of looking at scenarios where we're going to have to run 8,000, maybe 10,000, maybe, heaven forbid, even higher than that, tests per day to counter this pandemic. So we have all these positives. We are now we're now monitoring them for variants of concern. You know, viral evolution is inevitable, and particularly in RNA viruses, you're getting variation all the time. Most of it's inconsequential, but a few changes will increase the fitness and therefore be selected for an increase. What we have with COVID, SARS-CoV-2, essentially you've got an infinite population size that's continually generating variations. And the current selection pressure has been for increased transmissibility. And that's what we've seen with the alpha, gamma, delta, multiple times, multiple parts of the world. There have been convergent evolution on a number of base changes that have resulted in increased infectivity. Now, with the rollout of vaccines, that is placing a very steep selection pressure on the virus to render vaccines less effective. And I and others, you've seen Dr. Fauci say this, increased virulence is probably inevitable. And some predict that vaccines will be ineffective within 12 months. What we need to do is to try and minimize what I call the boom and bust cycle, and I'll explain a little bit more in the next slide. And so you can always think of it, what we're currently doing is only practice for when a variant appears that's not controlled by the vaccines. The equipment used at UCD was actually designed for SNP genotyping in the ag biotech sector. So it made very good sense to transition or use these machines as well to develop SNP assays, to monitor for variants in SARS-CoV-2. So we've been genotyping all positives, and within a few days, we share the results with the county health officer. So what do I mean with the boom and bust cycle? This is something that people have said to me, it must have been a big transition to start working on SARS-CoV-2. My response is, I've actually been working on pandemics for nearly 40 years, but they've been plant pandemics. But there's a lot of parallels between what happens in agriculture and what's happening with SARS-CoV-2. And we have what's called in plant pathology, the boom and bust cycle, where the breeders will generate a resistant cultivar by breeding. That puts a very steep selection pressure on the pathogen population to change, and then you get the bust. And we go round and round the cycle. And really, in plant breeding and agriculture, the question is how fast you go around this cycle. And much of our effort is designed to slow this cycle down and shift the equilibrium over to the right. I think there's a lot of parallels of how we should be countering this pandemic from this. So in terms of monitoring for new strains of SARS-CoV-2, I say the virus is continually evolving, though actually slower than some other RNA viruses. We have current variants of concern, particularly the alpha, beta, gamma, and delta, and they're more contagious. For example, there is convergent evolution, there are particular changes that render the S protein, is the protein on the outside of the virus, for binding to the ACE2 receptor. Delta is increasing rapidly in prevalence and pandemic severity. Now, alpha was actually first identified by detection of the S gene 6970 deletion, because many assays were relying on detecting this region of the S gene. So when you didn't see that, but the other parts of the virus were detected, that sort of raised a red flag and said we have a variant. It isn't the case with Delta. It's got other mutations. Now, the Davis test relies on detecting what we call N1 and N2 genes. These are not affected by any mutations that we know about that are present in the US. But we certainly monitor sequences of variants globally that could affect our test. So the CDC has come up with sort of 3 levels of classes of variants. There are variants of interest that they monitor. There are currently 6. And these are ones that there are changes in the sequence that we think could be consequential, but it's not necessarily proven. There are variants of concern, of which there are currently 4. And there is evidence for increased transmissibility or more severe disease and significant reduction in neutralization antibodies. So these are something that people are worried about. And then the third category is a variant of high consequence, where there's clear evidence that is consequence to the patient, the infected individual. And currently, there are no variants of high consequence, but certainly they're watching out for them. I say there are convergent evolution that's going on, and many of the mutations that have been picked up in variants of concern and variants of interest cluster into the S gene. That's the gene in orange here, pointer, and it's encoding what's shown in red on this graphic here on the outside of the virus. And it affects the binding of this protein on the outside of the virus to the ACE2 receptor that helps the virus get into the human cell. Now, the increased infectivity of Delta is really quite dramatic. This is data from last week or so from Public Health England. If people are interested in really knowing what's going on with the virus, I encourage you to go to their site. They have a tremendous amount of information. This is a graphic from there. And what this is showing in purple, that the rate of takeoff of the cumulative cases of Delta is greater than anything we've seen with the other variants of interest or concern. They don't have information for Delta because the data wasn't being collected when Alpha really took off. So much of the monitoring that's gone on has been based on sequencing. Sequencing, however, is expensive, it's slow, and basically generates information of historical interest, in my opinion. It's not really actionable. In the ag biotech community, they've been doing SNP genotyping very, very fast, cheap, and in high throughput. So this table kind of contrasts the genotyping versus sequencing. Genotyping is faster, it's cheaper. It's not very fussy about template. Anything you can PCR, you can feed into it. You can work with very low complexity machines. Turnaround is faster. Data analysis is easy. However, it is complementary to sequencing. It will not discover new sequence variants. So you still need to sequence, but you can do high throughput monitoring of the majority of your population with genotyping, and then be more selective in what you sequence. And now Public Health England actually runs genotyping first and sequencing second. So comparing SNP genotyping with sequencing, as I say, it's rapid. We can use diverse PCR-able templates, and basically we use residuals from the primary SARS-CoV-2 tests. You don't actually need to extract RNA, though it does help. It can be run on any qPCR machine. And in fact, if you don't have a qPCR machine, you can use endpoint assays. So anyone with a PCR machine and a plate reader, a fluorescent plate reader, can get data. but as I said before, it's complementary to sequencing. We still need sequencing to generate the information on which to base our SNP assays. Another important point, it doesn't compete with the supply chain for sequencing. There are definitely regulatory challenges. It's not FDA authorized, and we're a long way from having a clear certified SNP test that we can relate back to the patient. So all of this work is done for public health, not as a diagnostic that goes into the public health record of an individual. I think this is going to be a challenge if, when we inevitably do get variants that have direct consequences to treatments, we're not close to being able to plug that into the clinical diagnostics. So here's the workflow again, but what we can do is then, if it's raw, it's got a low CQ saliva, we can just plug it in at this place. If it's a high CQ saliva, or the residuals from a Bionax Now card or Panther swabs, we can extract the RNA and we can just put it into the cycle. So we didn't need any more equipment really, except to help us with the RNA extraction. Now, the SNP assay relies on very similar to the primary test. So the primary test detects the sequence, it's just got one probe. And what we mean by a probe is it has a short oligonucleotide with a fluorescent attached to one base, but a quencher on the other end of the oligonucleotide probe. And when the quencher is close to the fluorescence molecule, you don't get any fluorescence. But when it hybridizes to the template, and then there's amplification in the PCR reaction, the probe is digested and the fluorescent moiety and the quencher are separated, and therefore you get fluorescence, and it's quantitative. Now, in a SNP assay, you have two probes, match and a mismatch probe with different fluorochromes, and therefore it's basically a match-mismatch assay, and you look for the relative amounts of fluorescence in each case. And we can design probes for different sites in the virus, and that's what we've done in collaboration with LGC. We've got 25 assays that are currently working, and 12 of which are used routinely for genotyping. And you can see that the different variants here have a different pattern of nucleotide changes, and from that we can work out what variant is in the sample. What it looks like on the qPCR machine, you can see the trace and the endpoint value. Here you've got the wild type in blue, and in red, these were alpha. And you can see you've got very clear distinction in the amount of the ratio of one fluorescent moiety to the other. And if we go to the next one, this is a different nucleotide SNP assay. You can see here, this was a recent production sample, almost entirely all delta. The four blue ones here were actually controls. So we have, of our positives at times, 100% delta. If you don't want to use qPCR, these are what the endpoint assays will look like. Basically, they just take out the tracks, you're just looking at the endpoint. And to get this, all you need to have is a conventional PCR machine and a fluorescent plate reader. But at Davis, we're using qPCR, as I showed in the previous traces. So currently, we have a variety of inputs, raw saliva, swab samples, or extracted DNA. We're working with positive samples from both symptomatic and asymptomatic individuals. We look at 12 loci, and we can get informative data within a few days of sample collection if the CQ is low enough. Things that are of interest, we then take forward and sequence validate. But because we've seen such an upsurge in positives, and also it gets diminishing returns of just sequencing yet another sample of Delta. So we're focusing on vaccine escapes and unusual non-Delta samples. we report these results to the Yolo County Health Officer, and she's very welcoming of this data. Some of her fellow county health officers are somewhat envious, and at her behest, we've expanded to other counties, and particularly Nevada and Amador at the moment. An example of how this data was used, we were able to very quickly follow up. We had a variant. Here's the sample like this, and it was quite clear then that this actually matched B156, which is beta, and you can see the sample was collected on the 26th of March. We genotyped it 2 days later, and then we could report it. It was sequence validated subsequently. Looking at the data from what we're seeing currently, here is the takeoff of the Delta variant in green. You can see that we had a number of variants. This little yellow bump was the bump we got with Alpha. We did first detect Delta back in the beginning of April, but it kind of came and went, came and went, came and went. But then you can see when California opened up, and it just took off. And that's about as linear as you can get. We're seeing more positives now per day than we've ever seen. This is not good news. And this is in a county showing fairly high vaccination rates. So you've got a message that just because you're vaccinated, it doesn't mean that you can't get infected. This table here, if you want to follow the variants, is basically a table that displays the data I just showed graphically. And you can see color-wise that B117 or Alpha did have a bit of a peak back in April. And you can see here's Delta that came and went. But then you can see this really takes off. And we very rarely see any Alpha. And you can see over to the right, the number of positive symptomatic samples versus asymptomatic. You can see the testing, people kind of dropped off. And then it's now 2.5 times what it was. So yes, encourage people to test, and people are responding. With the sequence, we can run it against all the known sequences that are publicly available with a program called Usher. Jonathan Eisen's been doing this, and you can put it into clusters. And so far of the sequences for Delta that we have in the Davis in Yolo County, we've got about 50 clusters. The largest has got 34 samples in it, spread over several weeks. So what this is showing, with the number of clusters show we're getting multiple seeds, migration into the population, the community, but then we're clearly getting community transmission. And you can see that some of these individuals' vaccines escaped. So, this allows us to monitor or inform that we are getting community transmission, and it is coming from vaccine escapes as well. Now, we're not going to solve the problem just by, in California, Yolo County, US. We've got to grapple with this globally. And looking forward, I think genotyping can really help surveillance on a global scale, particularly in areas where routine sequencing is not feasible. And I'm collaborating with people. We really want to have complementary high throughput genotyping in many areas around the world so we can monitor for new variants as they come. We do need regulatory acceptance for public health actions. And particularly, I think, once we get it actionable, whether we do it or other labs, clear certified labs, used SNP genotyping for patient interventions. Now, so far, I've been able to get, we're getting the tests out to people in Colombia, Senegal, Tanzania, and Cambodia. If you know of locations that are doing testing that have the capacity to run qPCR or PCR and would be interested in an early access program, please let me know. I think it's incredibly important that we push this out. The importance of getting tested and surveillance after vaccination. As I said, just because you're vaccinated, it doesn't mean you won't get infected. And it's critically important to monitor for variants. The vaccines are highly effective against hospitalizations and death due to the current variants. The vaccines may have reduced efficacy. You can still get infected, though at a much lower rate than if you're not vaccinated. And remember that 40% of infections are asymptomatic. So it's possible that vaccinated and unvaccinated individuals who are asymptomatic can have very high viral loads. I'll show that in the next slide. So, and we also, infections from vaccinated individuals are clearly happening. So it's very important to continue testing after vaccination, and we require monitoring to test and genotyping results in the context of vaccination records. So, we, and it's sort of a national effort, are seeking vaccine escapes to see whether there are any critical mutations that lead to this. So people have asked, with the vaccine escapes, what's the average viral load? So CQ is inversely related. CQ is the number of cycles you need to run on the PCR machine before you can detect it. So a high number means a low viral load, and a low number means a high viral load. And what's interesting in this data, you can see in blue that the average CQ is slightly higher than with Delta, but also that the vaccinated individuals, well, it may be trending slightly higher in the vaccinated individuals. It's still pretty similar. And there are individuals here with very high viral loads. Now, it doesn't mean that we're detecting virus here. It doesn't mean it's quite the same infectivity, because you need to do studies with neutralizing antibodies. But what it does, it really says you can be asymptomatic, you can have a lot of virus, and we know you can get transmission. So yeah, you need to test, and just because you're vaccinated, it doesn't mean that you're not potentially a liability to the population. So to wrap up, this has been a tremendous team effort. These individuals here are just those individuals associated with genotyping and sequencing. There's about 2 to 3 times this number of people associated with testing. And with that, I will wrap up.
Thank you so much, Richard. I can remember that day when we were all talking with Provost Kogan, and you made the case for the investment, and the investment was made. And we are so grateful for your passion and your commitment. And that data is really sobering, and really speaks to the need for testing for everyone. With that, I'm going to pivot to another colleague who has been just incredible in partnering with Healthy Davis Together, the School of Medicine, the Office of Research. I know with your group, Dr. Brad Pollack. Brad is a national figure like yourself. He's a professor of epidemiology and chair and associate dean of the Department of Public Health Sciences at the UC Davis School of Medicine. He wears so many hats, I probably can't list them all, but he is a leading researcher in epidemiology and control of childhood and adolescent cancers. And now, thanks to the pandemic, he, like you, are a leading researcher in COVID and how it spreads. His work with Healthy Davis Together has been a game changer for UC Davis, for Yolo County, for really the country. And your work, Richard and Brad's work, and the work of so many has made such a tremendous impact on the lives of our community. Brad serves on the board of the University of California Global Health Institute, and he is representing UC Davis in global health interests across the system. Brad, on a personal level, you have been a leader that has been always available through this entire pandemic, and your voice is always one of extreme reason, and we are so grateful to you. I'm going to turn this over to you.
Thanks a lot, Allison. Appreciate that. And it's while the pandemic's not fun, it's been fun to work with folks like Richard and a lot of other colleagues that we wouldn't have had the opportunity to do were it not for the pandemic. So thanks. I'm going to talk a little bit about the Healthy Davis Together project. I'll be saying a couple things here that represent my own opinions, not necessarily those of all of my project colleagues or the university. I'll try to delineate those. But first I want to start, as an epidemiologist, you got to start with the numbers here, right? So this was a snapshot of the Hopkins dashboard from yesterday. And, what we're at right now is globally, over 4.3 million deaths from SARS-CoV-2 and about 619,000 in the US. So that's sobering. Last year, COVID-19 became the third most common cause of death in the US. So this doesn't happen every day, believe me. These sorts of things shift around over decades, but this was very dramatic. And if you just want to see the magnitude, this is looking at the daily trends. This is off the CDC website for cases identified in the US here. And again, we know we thought we had our peak in January, certainly in California, mid-January, but look what's going on. We're getting the other peak, and which is due to the Delta surge right now. So that's a snapshot of where things are now. But I want to take you back a little ways to early last summer. We had developed our campus ready plan. We were doing a lot of preparation to get students to come back in fall of last year. And we had the opportunity to think a little bit outside the box and put together a concept to address the pandemic, not just for the campus, but actually looking at the Davis community to extend the campus ready plan into the community. And we came up with Healthy Davis Together. So that's you've seen that logo around town, I'm sure. We launched this officially in September, but that was after a good 3.5 months of planning. And the goals of Healthy Davis Together are pretty straightforward here. Obviously, they're to lower the burden of COVID in the Davis community, but also at the time we envisioned this to facilitate a coordinated gradual return of regular activities in the city, and in particular, reintegrating students back into Davis. Again, think about the thought processes back last summer, before last summer. It was basically that, oh my gosh, you're hearing about all these college campuses that had started back up. There are outbreaks there. These students are coming from all over the country. They're going to bring contagion into our town and infect the townspeople. So we kind of thought about that last summer about what we might be able to do in the community setting to alleviate some of those fears. And the other purpose of this is to try to identify replicable and scalable approaches that we can generalize to other communities, perhaps other college communities, but other communities in general. A longer range goal is to really try to establish lasting public health infrastructure to promote health in the community. And that's to do that well beyond the pandemic. We thought the pandemic would be over by now. It's not, but eventually it will be over. All epidemics, all pandemics end. They have an end, and it will happen. But we are concerned about all the efforts that we've made and how some of these can be leveraged into providing lasting infrastructure. So this is a schema for the entire project here. And I just want to kind of walk you through it very quickly. On the left-hand side, you'll see there's two major themes of this. And what we are trying to do is synergistically combine epidemiology with a number of aspects of epidemiology with health behavior change. And it's the synergistic combination that we thought was going to get us over the hump to really be effective. And if you see within epidemiology, we identified a whole bunch of things that we called approaches. So that would include testing, and again, teaming up with Richard and the Genome Center for that, contact tracing, exposure notification, isolation, quarantine. That's all kind of part of that triad of the infectious disease control, but adding some other things in here, including modeling and forecasting, environmental monitoring, and so on. For the health behavior change side of things, this is really health education, developing mass communications, developing incentives, partnering with different organizations, different individuals. We partner with the business community in Davis. And of course, the target or the beneficiaries really include everybody and all organizations, all people in Davis. That was the initial scope of the project. Because of the complexity of this project, we also identified a number of cores shown on the bottom here. These were kind of supporting cores to help all aspects of the overall project. So just to walk you through the epidemiology theme here, Richard's presented the approach we've done using the mass asymptomatic PCR testing platform, which has been incredible. And I remember the talks go back before late last summer. We were talking in May, I believe, Ken Burtis and I meeting with Richard and trying to twist a few arms to put in place this really high throughput platform that turned out to be, the best insight and decisions that we've made here, I think. So we deployed this obviously in September. It went live when we had the students move in last summer. And then we extended this out in mid-November to the Davis community. In addition, we also knew, because cases were going to be identified, and the county was very strapped at the time. In fact, when we talked to them back in April of last year, sometimes some of the cases they were trying to get a hold of were at least 8 days out from when they got sampled. And that had a lot to do with the slower testing technology, as well as being understaffed there. So we actually came in at UC Davis and assisted with case identification on our campus, and certainly then extending out to the Davis community, and then becoming official contact tracers, working with the county health department. The testing and the contact tracing combined really reduce the delay between case identification and then triage to appropriate isolation or to quarantine for high-risk contacts. That gets the number of people out there away from infecting other folks. And not just to get them in isolation and quarantine, but also to think not only about the housing, but also food, social support services. And in some cases, we were able to arrange wage replacement. So these are important to not set up a lot of barriers. And that, of course, on the campus, it was relatively straightforward because we had a new dorm set of buildings we built. So we were able to allocate those for triage, allocate them for isolation and quarantine. But in the city, we partnered with several of the commercial enterprises to provide those types of spaces. So in addition to that, part of the epidemiologic approach is also to be doing environmental monitoring. This is showing you some of Heather Bishel's work. Heather's led our wastewater monitoring that's been part of Healthy Davis Together here. And where we are doing monitoring, not just at the treatment plant, there's one treatment plant for the city of Davis, but actually doing this at the sub-sewer shed level. So we're able to detect virus and wastewater at the neighborhood level, and trying to do some very pioneering work there. In addition to wastewater, David Coyle's been running our air filter monitoring program here, where we've actually set up indoor air filters in a number of the Davis Joint Unified School classrooms, and looking to see how that will fit into the overall prevention scheme, in addition to doing some contact surface monitoring. And of course, vaccination, which was a pipe dream when we started this project, when we were planning it out, became a reality in December when we had our first EUA approval of the Pfizer vaccine. But we were planning for this all along. And as the vaccine program rolled out, or even before we were able to get vaccines out, we contacted the manufacturers, and we contacted one of President Biden's transition team leaders for the COVID-19 transition team to see if we can get a special allocation to do a pilot study in Davis, and no dice. None of the vaccine was available to us. And that was because of just the way that the vaccines were developed. So we weren't able to do that, but we did see early on that the vaccine distribution was going to be first going out to healthcare provider organizations. So we actually pulled together the healthcare provider organizations, the large ones in Yolo County, along with representatives from retail pharmacy chains, and tried to get other folks that were going to be involved in vaccination together to see how we could help. And one of the things we did was we partnered with our federally qualified health center in Davis, Communicare, and with Yolo County Public Health to set up a number of vaccine sites across the county, in particular targeting the harder to reach and vulnerable populations. We deployed mobile vans. We've done door-to-door vaccinations for the Project Room Key unsheltered folks, and partnered with a number of community-based organizations, and with the Orlay grant, that's our NIH grant here, to reach out specifically to migrant farm workers, where we've achieved very, very high vaccination rates in Yolo County for that population. The health behavior change side of things really involves mass communications using all sorts of different types of media. You've probably seen the colors and these logos. You're probably sick of it if you live in Davis, but we've barraged the community with a lot of advertising here. We established the Aggie Public Health Ambassadors Program, which is made up of undergraduates about 200 or so that have been in paid positions. They were first deployed on the campus and then have rolled out and have been deployed since Thanksgiving time last year in the city and the farmers market in the downtown Davis area. In addition, we established a business partnership program. And then you've seen we had a number of really neat incentives here. We actually funded the $75 staycation spring break thing, the Halloween event that we had. And so that's been part of the health behavior change side to change behaviors. And I'll say, I'm not going to present the data today, but we have been doing surveys every 2 months of 600 or more individuals from Davis and from surrounding communities to see how some of this messaging has actually penetrated in terms of their understanding certain personal health behaviors as well. Just to show you some very, very high level results here. Well, Richard showed you one of the figures here. The left-hand panel is showing you the case positivity rate over the different days, going back from when we started Healthy Davis Together in mid-November forward to a couple days ago. And you can see again, this spike in Davis now where we have exceeded our highest case rates per day. That's unfortunate. We're dealing with the Delta variant right now. On the right side, though, this is very telling. This is kind of doing a gross comparison of case positivity rates. The bottom two lines represent the UC Davis campus itself, and then Healthy Davis Together is a line right above it. The third line up is Yolo County, excluding Davis, and then the top line is the state of California. And you can see that we really did keep much lower rates, but now even as things are spiking up here, it looks like Davis is doing much better than the county in general and California as well. And this is just showing you again, sort of the testing volume, which tended to ebb. In fact, when we had vaccinations being rolled out, people stopped getting tested. And in fact, it wasn't helpful, in my opinion, that the CDC 3 months ago said, you got fully vaccinated, you can hug your grandkids, but you don't need to get tested anymore unless you're symptomatic. And when I saw that, I couldn't believe that was their advice. And the next day I was on a panel with Michael Minna, who's been a big proponent of testing. And he said he almost fell off his chair when he saw that. We in Davis have messaged the opposite of that. The chancellor even came out with a strong Friday message about the fact we want to keep our asymptomatic testing up, whether you're vaccinated or not. And now you can see the utility, as Richard explained, with the shift in the variant. So it's really, really important. But some of the other results we've had here, we've identified 2,200 people in Davis who were positive among 54,000 individuals who've been tested. Not always every week, but certainly just overall a lot of testing here. I identified people that were considered high-risk contacts that could have been exposed and so on. And we actually launched over 70 different testing sites. Some of these have been smaller, as well as our community testing sites for most of our testing. And then for vaccinations, again, we were trying to find our middle ground here, what we could do to help. We weren't going to replace the healthcare provider organizations, but we've really worked by setting up some pop-up vaccination clinics, mobile operations, and so on. And actually, our doses that we've delivered in our program have really been allocated to about 40% of Latinx residents of the county, as opposed to the overall vaccinations, which is about 25% of the county doses. So that's kind of just a quick high-level view of the results. I want to also point out this slide is showing you Yolo County as a whole. And what's really important is we've taken the approach of using our own data to actually more precisely target where we want to do our interventions. This is called precision public health. This map shows you some of the analyses we've done where we have set up about targeting for both extended testing as well as vaccination. So that's a really important attribute here. And just as an example, if you think about wastewater monitoring, again, Heather's group has done this and partnered with the city of Davis now, but using some of that maps and the trends that we're looking at here, we were able to identify a couple of neighborhoods that kind of lit up, and that led to putting door hangers on people's doors in selected neighborhoods. And the door hanger had instructions and how to get tested easily. And we even had a couple of these that had incentives tied to them. So that's an example of using precision approaches. The other key part of this project is knowledge sharing and dissemination. And we have an entire core that we work on where we've actually put together a number of what we call white papers, which are kind of the very detailed methods how to do things. We've put together playbooks, which are sort of designed to be distributed out to policymakers. We did one, for example, with safe school reopenings here, and those are available publicly, and they've been picked up by other school districts from around the country here. And of course, we're working on the scholarly publications. Got our first abstract accepted to a national meeting for an overview of the results coming up next month. And again, the other thing is a lot of us have been involved in doing some media events, trying to push this out, push this information out. And again, we want to see what we've kind of learned used. Not as definitively evaluated as we'd like, but we're in the middle of a pandemic. We can't wait 2 or 3 years for all the publications to come out. So we've been using other media as well as white papers, non-peer reviewed, to get that and disseminate information out. So I want to say that I think the innovation of the project here has really been the synergistic combination of both the epidemiologic infectious disease control, which you need to do. Other campuses around the country have done that, but combining that with the health behavior change. As we say in epidemiology, you can never test your way out of an epidemic. And that means that there's something more than just testing involved. And of course, it involves behavior. We also extended the bubble that we developed with our Campus Ready plan to the whole Davis community. And now in the second phase, we've got a new moniker of Healthy Yellow together. We've just diffused out into the K through 12 schools and other aspects of Yellow County, other school districts. And as I said, we're using our own data to do a precision public health guided approaches to how we do our interventions here. The last thing, as I mentioned at the beginning, we're trying to identify effective, replicable, and scalable solutions here that can be given to other communities, and also to help us build our lasting public health infrastructure. I think some of the enduring benefits that we're just observing now is we've really seen a strengthening of the collaborations we have between the university and the city of Davis, the county, and also with some private entities that have worked with us here, including CBOs and Fairly Qualified Health Center. The environmental monitoring, it's not a solution for or a replacement for individual testing, but it's going to be an important adjunct going forward for doing pathogen and emerging pathogen surveillance. And I think the other thing is we certainly increased the public health literacy of the community, at least in Davis, which has impacted health behaviors here. And I think also, I was at our academic public health meeting a few months ago, and Tom Frieden was our guest, and Tom was asked the question, what do we need to do to prevent the next pandemic? He said, labs and surveillance, kind of like Dustin Hoffman being told plastics for what career to go in The Graduate, the movie. It was labs and surveillance. And we clearly see the need for heightened community testing. I think, again, a lot of what we're able to do now, and a lot of Richard's contributions here, really depend on being able to test larger numbers of individuals, and especially to do that in a relatively fixed community, not exactly fixed, provides a lot of power. I think thinking a little bit about what's going on, the CDC website here has listed a few things here about surveillance here, and with regard to variants here, talking about doing this to inform outbreak investigations and national trends. And basically, the statement, viral mutations and variants in the US are routinely monitored through sequence-based surveillance, laboratory studies, and epidemiologic investigations. I think they're routinely monitored in some places, but the question you have to ask is who is being monitored, which populations, are they representative of the overall general population? If not, why not? And again, I think that points to the utility of having Davis be involved in this and doing the testing that we have done. It's going to be very important to contribute to the overall scientific base of how you handle these pandemics. A couple other things here. I think the next steps here, and the CDC has been a little bit slow to get to this, but it's really, we need to develop some strategies or a strategy to monitor the spread of variants of concern in a better way. In a true population-based surveillance manner where we can leverage our testing centers. Well, we have the big testing center right now, but also we're not getting everybody in Davis or everybody in Yellow County tested. And I think we need to develop appropriate probabilistic sampling schemes like you do in any kind of normal sentinel surveillance monitoring system. We need to be able to connect up labs and coordinate testing and collection. And I think the other thing we saw that was incredibly poorly done is the infrastructure for IT and informatics. It was horrible. And the state system was horrible. It got the last CDPH director fired, partially. So I think this is something we need to pay attention to with appropriate interoperability across labs and other sources for getting information in, as well as ensuring that we have confidentiality, security, and privacy. And I think, overall, not just for variants of concern for this pandemic, but just having sentinel surveillance systems that are set up to be sensitive and practical for emerging pathogens is important. I think environmental monitoring will play an increasingly important role. Maybe other animal species, not necessarily for this particular pandemic, but I know that's the work of John Amazad and a lot of our colleagues at the One Health Institute, is to really look at how we can use sentinel animal species, for example, as well as other ways of linking together information about emerging pathogens. So just a couple concluding statements. This will get more into my opinion rather than reflecting my colleagues I'm working closely with. But, I think we've seen like never before where health disparities, health equity, and social injustice really came to rise up. and explain a lot of the problems we've had with this pandemic. You remember the death rates in New Orleans that were so linked to the African-American population there, which had much higher comorbidities. This has never been brought more to the front than this pandemic. And to see that we had civil unrest matrixed on top of a pandemic, well, my group, the UC group I've been chairing, many of us wrote this article that got published in Public Health Reports about recommendations for demonstrators and law enforcement agencies during civil protests. Who would have thought we'd write an article like this? I mean, it was just almost surreal. I think also we've seen the effects of politicizing health. We've seen information being twisted, where we've distributed misinformation and disinformation, where there's purposeful deflection and diffusion. We have had incredibly low levels of public health literacy in this country. When you talked to a person a year ago and said, we told them don't wear a mask because we don't have the evidence that it really is protective, and it may make people feel more aggressive about getting close to other folks. And then a few months later, we now recommend you wear a mask to prevent transmission to others. And a few months after that, we find out, well, there's some protection for the wearer. Well, you go back to the same person on the street and say, can you please wear a mask? What's wrong with you guys? First you told me not to wear a mask, and now you tell me to wear a mask. What's wrong with you guys? Well, that's a reflection of the low level of scientific literacy in this country. And if people understood how evidence develops and how we work on things, wouldn't have had, I think, that same reaction there. And again, we've seen how incredibly hard it is to get many people to do, quote unquote, the right thing. On the plus side, we've really, I think, successfully leveraged what I consider a highly interactive transdisciplinary approach to everything dealing with this pandemic. It includes characterizing the virus and its impacts, looking at the effectiveness of non-pharmacologic interventions, but developing the vaccines, incredible, and refining modeling and forecasting approaches. Although again, the big problem we're having there is data. We just do not have good data infrastructure to be able to do this in as efficient a way as we'd like to do it. So I think, we were very ill prepared for this pandemic. There was a decades-long decline in support for public health infrastructure. Starting when I started graduate school, I had in 1979 at UCLA, I had a U.S. Public Health Service manpower grant to cover my tuition and a stipend. Reagan got elected in 1980. He removed all those overnight. Those were not. They were just cut. And to my mind, personally, that's when I saw the erosion of public health infrastructure investment. Maybe that's changing now. We hope it will. We certainly have, from our work, preliminary evidence that community approaches to control of communicable diseases can be effective. And I'd say that while biology is a predominant driver of epidemics, never underestimate the impact of human behavior. If you think about this, that's the wild card here. We would not be talking about masking mandates today if individual behaviors were predictable and rational, not rationale, but rational. Quashing a pandemic, obviously involves more than merely developing a highly effective preventive measure like a vaccine. We're seeing that right now. So I think our challenge as we go forward here is to sort of overcome some of those obstacles. But I want to say we really have leveraged basic biologic discovery, clinical research, and population-based approaches to address the pandemic here in Davis. It's exciting to be working in this environment here. And I just want to do a couple shout outs or acknowledgements here. Our executive committee, we were meeting 3 times a week, with a lot of other side meetings beyond that, but it includes the folks you see listed here. Actually, Ken Burtis was our first chief operating officer, and then Todd's taken over for Ken. In addition to that, we have a phenomenal advisory committee. It's chaired by the provost and by the city manager, and a long list of characters and wonderful people from the community representing different constituencies have worked on that. And we also have a really interesting, great set of experts that we brought on as our external advisory committee members. Lastly, I want to really thank the approach leaders and our core leaders, all those things you saw in that schema. There's a lot of people behind all of those things, and so many staff who've worked so hard to make this successful. Finally, and very importantly, I want to thank the chancellor, Gary. This is Gary's project. Gary made this project possible. We're very, very fortunate we had the opportunity to do something that we think will be very impactful going forward. And I will wrap it up right there. Thank you very much.
Thank you so much, Dr. Pollack. Amazing presentation. We're going to pivot quickly to Dr. Bart Weimer, Chair and Professor of Population Health and Reproduction at the UC Davis School of Veterinary Medicine. Dr. Weimer is a leading researcher in food health, microbiome, bacterial population genomics, and he also leads the 100K pathogen genome sequencing project, enabling metagenetic solution. So welcome, Dr. Weimer. We're excited to have you.
Thanks very much. I hope you can hear me okay and you can see the slides.
Excellent. Yes.
Very good. Thank you very much for the introduction. And I want to thank the student who's working on this, primarily Darwin Bandoy. He's a veterinarian from the Philippines who's come to Davis to get his PhD in population genomics. And so, like others, especially Richard, I want to say, you know, I've been working on this really not in SARS, COVID, but in other organisms for a very long time. And so I wanted to show you a little bit about some of the impact of the genes and some of the impact of the mutations that have biological consequence. So there's always a debate about whether it's more virulent or more transmissible. And so we've been looking at population genomic approaches to try and figure that out. But most people have looked at the spike protein because that's the protein that interacts with your cell. There are mutations around the other parts of the virus. As Richard talked about, they're monitoring some of those mutations. And he also said you can't find new mutations if you don't sequence them. So a monitoring program using PCR is fantastic. Sequencing is really going to find those new mutations. And you can see that outside of the spike protein, there's a couple of other genes where you get increased transmissibility of the virus. So the one thing to kind of think about is, you know, over the outbreak time period, which we all thought would be over by now, when we first started seeing cases, the age, you know, we were really looking at elderly folks getting sick, and today we're not. We're looking at younger populations. So the far right part of the slide, we just published this with some colleagues in the UK, looking at, you know, what's the average age or what's the age demographic of folks getting sick today, and it's much younger than it was when we first started the pandemic. And I think I'll go ahead and say this, even though there's a lot of people, as one of those people who had a breakthrough infection, I got infected from a 14-month-old child, and as did 2 other people in the household. And we were all vaccinated. We had Moderna and we had Pfizer vaccine. So it is occurring. I'm one of those 2,200 that Richard just showed you a few minutes ago for the new case of this was recent. I got it last month. So it is happening, you know, of an N of 4. You do see it, it will happen. And I'm going to show you the underlying genomic reasons that we keep getting bursts in outbreaks. So one of the ways that we've been trying to think about this, and you heard it in both previous speakers, is that the infrastructure for data analysis, we were not prepared for the onslaught of data that we were going to have. In genomics, we typically, you know, put things into trees like Richard showed, and that works very well when you have about less than 1,000 genomes or less than 1,000 samples. Once you get over 1,000 samples and you start putting it into trees and lineages, it's incredibly difficult to bin it and make sense of it from a public health perspective. And so my group has been working on not only in SARS, but also in bacterial systems where you have high mutation rates, not as high as SARS-CoV-2, but higher than normal. And it's all based on the mutation rate that you're going to see these spikes in transmissibility and spike in outbreak. So when you really look at it from a global perspective, there's a theory that we can talk about both from epidemiological perspective that you just heard about, but also from a genomic mutation and genomic evolution perspective. So Richard used a lot of words that were spot on, you know, like convergent evolution and selection and the mutation rate. So I want to show you some global perspective for just a minute. Not that you haven't seen these data for a while, but on the top left is the UK, which is where we primarily get most of our data. The reason is because the UK was a very, very early adopter of creating a sequencing consortium called COG, which then they publicly released all of their data. Unfortunately, our public health system has not done that to a large degree. So there might be surveillance occurring, but a lot of the data are not available. So you can see over time from June of 2020 to June of 2021 in the UK, they really had 3 waves of different variants that were causing disease. Unlike the unlike the US, where we really had, you know, multiple variants of interest or multiple variants all circulating in the population generally. And then again, opposed to Brazil in the lower right, where they had essentially 1 or 2 variants, and Canada was more of a mix like the US. And they had something called EU2 that's not really spiked anywhere else other than Canada. So that actually is showing a term that's known in ecology and in evolution. It's called clonal interference. And as you plot changes in the prevalence of a genotype over time, so you can look at a longitudinal prevalence, we now are at a point in the pandemic, unfortunately, that we can start applying evolutionary genetics and evolutionary population genetics to the data sets that are now widely available. And the infrastructure to analyze those data are really poorly developed, in all honesty, both locally, meaning in the US, but also then globally. So we're all struggling to plot something other than what these plots show. And the way we think to do that is to start using emerging genomic variation in each individual gene or each individual variant, along with epidemiological principles that are well trodden. And so you can get beneficial mutations that are selected, meaning beneficial in this case, perhaps negatively stated, is causes an infection. So you see on this, one thing that you all want to point out is you see that, whoops, I'm so sorry. You see that, you know, variants come and go. They rise and fall. They emerge and they go extinct. The problem is that extinction doesn't necessarily mean they're totally gone. They're just below the detection limit of the surveillance system that's working in whatever location you're in. So they can reemerge, which we're seeing. So this is by state. So when you look at California in the upper left corner, highly dominated by Delta right now. There are a couple of other variants that are still circulating in this yellow color, which is a 21H. Florida is a different shape, different variants. New York is a different variant. So what I'm getting at is that while we talk about it as a US pandemic and a California pandemic or epidemic, it's not. It's unique to each location, which is what you just heard. By looking at Davis alone, we're making very strategic local decisions. And by not segregating out and changing epidemiological and genomic surveillance in each location, you're really gonna undermine any progress that you could make because there's so darn many variants circulating. This is only a partial list of variants that are in circulation within the human population right now. So it's no wonder, unfortunately, that we keep seeing these spikes, but Delta is taking over at the moment. If you go back to April, that's definitely not true. It's Alpha and it's Beta and it's others. And you keep hearing about other variants that are coming into the country and moving around. So you don't have to wait for people to move around. You can get spontaneous mutations in a local setting, which then lead to another system like this. So what that I want to point to is I've showed this before, but actually almost about a year ago, I showed this, and it's very techie, I'm sorry, but it's a great way to, it brings out a point that I want to make. And the idea of clonal interference is you can use selection. And the selection that we're seeing is us, our immune system, and now the vaccine are selecting the variants that are going to persist into the future. So when you get vaccine breakthrough, then you have a new opportunity to have a new variant that can escape future vaccination and other vaccines. So you're not, as you know, as you just heard Brad talk about, you're not quote safe. The symptoms are much less severe, yes, but this clonal interference is really what's at play over time. And the longer you go, the more variants are going to emerge. But you have to have some amount of convergent evolution to maintain infectivity. So some lose the ability to infect and they go away, and some maintain and gain better infection capabilities and more severe symptomatic, and they then get selected in the population, especially if you're not vaccinated. I show this really for two reasons. One is to show that you can maintain them. Variants can be maintained in the population, or SNPs can be, variants can be persistent. They all will change, and that's what these crossover points are. But what I really like is this chaotic part in the lower right corner, showing that there's a lot of variants that are moving around and a lot of variants that are coming and going. And this happened to be when lockdown was instituted last year. We were on a very nice trajectory of selection of variants that were going to fix into the population, and we knew what we could deal with. When lockdown occurred, all of that went into chaos, and you get this stochastic variation within the genetic variability. And so then when you put a new selection piece in place called the vaccine, this pot of turmoil then starts to then progress into a selection curve. So while this is a very chaotic plot, it shows something very nice that's biologically informative and disease informative as well. So what we did is we published a paper last year, which I'll end the talk with, but this is our approach at the moment, where we're really talking about taking classical epidemiological cases and epidemiological curves that you just heard about, and whatever metric that you use, whether it's sequencing or whether it's PCR screening, we end up turning that into a variant epidemic curve, not a total or an average epidemic curve that you would typically see, but an epidemic curve like I showed you at the second slide, where you're seeing cases by each variant. By knowing those cases by each variant, you can turn it into the reproductive number, which we've all heard about by now, not the reproductive number that's an average of the overall pandemic or epidemic in the local case, but what's the reproductive number of each variant? And that can be informative. Now that we've got a year and a half worth of data, you can also then introduce a time variable and a time series analysis into this piece and say, wow, we now have in England, from England, we just did, I wrote 300,000 sequences. We did it in 500,000 sequences. And when you try to put 500,000 sequences into a tree, into a chart, it's outrageously difficult. And in fact, most people in the world now are moving away from phylogenetic trees and moving into variant tracking, variants of interest. So when you produce a variant reproductive number, then you can start talking about who's susceptible to that variant, what's happened to your social changes, whether it's pharmaceutical or behavioral. So this allows us to harness all this massive information that's being produced, because we're not going to quit. And that's the focus of the 100K Pathogen Genome Project, is that we're looking at population genomics in the microbial world and the viral world in a way that we've never, ever had the capability of doing before, because we have 500,000 sequences, not 5 or 10 or 20 or 50. Now we can look at really populations. Okay, so I want to go and tell you a little bit about what an R sub V looks like. So you know what the first wave, the second wave. This is what's happening in the wild type, and in the mutant in the second wave is dominated by 614G, but you also then get a change in the circulating viral variants that are going in each wave. So now not only is the first wave different by what the dominant variant was, but you're also getting a population of variants sitting behind it that are also changing, that can then become the next wave as you move on. And so we calculate the R value for each of those, and post-lockdown, the virulence actually went down, the R went down, the transmissibility went down, yet the number of cases went up. You have to ask why. So there's this whole problem about what we do. And we from a genomics perspective, we've got so darn many sequences that we're not using really in a wise way. Not because we don't want to, it's because the computational infrastructure is not there, and the overarching integration approaches are not yet well developed. They're emerging around the globe. Okay, so lockdown did its job. Lockdown actually changed the case rate. It also changed the variant population. And when the second wave came along, you got a whole new variant that caused a whole new outbreak number of cases. So, and the circulating variants changed in the second wave, which we wouldn't know if we don't surveil. So as the RV, or the transmissibility estimation, it changes in time. It's not a static constant. And that is a question of why not? Why doesn't it stay the same? It's the same mutation. So is it the underlying other mutations that are going on in the virus that are co-mutating at the same time? Yet to be described, yet to know. Is it behavioral issues that we don't know? We've got a new susceptible population. Is it age? Is it health? We don't know. There's probably some of both, all of those. But certainly the transmissibility over time of a single variant does change. And you can see that in both of these cases. And in the alpha, you can see it rose and fell. And this is, again, what you've been seeing in other presentations, but you're seeing the circulation of a variety of items, okay? So we've already shown you that, yes, the age is changing, the symptoms are starting to change. So what we wanted to do is not look retrospectively. We wanted to use the data from this longitudinal assessment of all that's been accumulated and try and be predictive and start trying to do a bit of modeling. Can we start predicting what's going to happen to the future so that we can then change our behavior in some way? So it's classical epidemiology. You start looking at the SIR model, or the susceptible, the infected, and the resistant. And this is with the wild type, you get about, in this particular scenario, you get about 6,000 cases. And then the resistant and the susceptible population change. Well, in the mutant, you get a much higher caseload. It's much more transmissible, much more infective. And so as you get a new mix of variants coming along that are circulating, it's a pot of mix that is, let's pick the next variant that can do something new by the selective approaches that we take, whether it's social interventions or pharmaceutical interventions or both. So as we start modeling each individual variant, you can start making a lot of conclusions. So on the right side of the screen is the case count. So the classical epidemiological approach, it was bumping along, and then it declined very quickly in early 2021. So the blue line we just turned that into a variant R, and we measured it, which is the blue line, and then we started using modeling, which is the red line. So the model that we're using is predicting very well what's going to happen over the entire time frame. So we said, okay, that's great. We were able to predict what happened when it went away. Can we predict one that is escaping vaccination? And sure enough, the answer is yes. We can see this curve on the right, which is the case count essentially. And then the curve on the left is the R for that variant and the predicted R of cases. So now we think we have a way that we can start looking at what's going to happen into the future. How can we take this population of data, assemble it all up, look at it in a really rigorous statistical way, and then decide, can we start estimating what we need to do proactively rather than retrospectively, because we're always in this reactive mode, which is what Brad sort of talked about and what CDC's sort of giving confusing communication on. Mask, don't mask, don't go ahead and hug your grandkids, don't hug your grandkids, and depending on vaccination. And then when the next really big question is, when do we need a booster? And what booster do we really need? What variant should we be making that booster against? And so this chart is really actually sort of saying to you, we believe we believe we can start predicting what's going to happen. Of course, we may get it wrong, but with the amount of data that we're using, 500,000 genomes and untold numbers of people over these cases, then I think we've got enough rigor in the math that we can start being very proactive and very accurate into the future rather than retrospectively. So this is something that we published about a year ago. Essentially, it was a prelude to what I just talked about, and we published this paper in showing how it would work very early in the pandemic. We started with 150 genomes. By the end of this paper, in the review process, we did 20,000 genomes, and the data I just showed you were based on 500,000 genomes. And if I drew a tree on those 500,000 genomes, I would not know how to interpret it, to be very honest with you. So we believe this is a bit of a roadmap of how to use the integration of population scale genomics and epidemiology and a bit of modeling, both classical modeling and machine learning and AI informed modeling that's underway, that we think we can do a pretty good job of in predicting what sort of variants are coming next, where are they going to be, and what kind of vaccination or behavioral process should we be changing to help mitigate the expansion of the outbreak. So just a couple of conclusions. We think that using population genetics and evolution is a really sound way to go. There's a lot of theory around it. There's a lot of, as we now know, sequencing and genotyping are great input data sets for these sorts of approaches. And surveillance is never going to go away. We're going to surveil this for a very long time. Epidemiological programs are built on surveillance systems, whether you're in humans or in animals. And the surveillance systems in animals are very well built, to be honest. And a lot of these sorts of coronaviruses are already surveilled in certain populations of livestock and in animal health. So unfortunately, what's on our side is we have very large numbers. The fortunate part is the large numbers are great. How we got them is the unfortunate part. And so new waves will continue. We will start seeing new waves and new emerging variants and new the list is only going to change because we still end up with this circulating population. It's not a monomorphic single variant outbreak. With a highly diverse organism and a highly mutable organism like an RNA virus, it only takes a little short time period to get mutations to occur. Even in bacteria, who are relatively slow mutators compared to this virus, you get new variants within a day. So it's very easy to get new variants, and it's very quick. And most infections, you're not infected with a single variant. While it's the dominant variant, it's not the only variant. So I would be interested eventually to see if Richard's been able to detect more than one variant in a single spit sample. That would be really interesting to know. Okay, so the last thing I want to talk about is just the ability to predict into the future. We think by using this approach, we think we can start predicting methodological approaches to limit the pandemic. Okay, very good. Thank you very much for your time and the invitation today, and look forward to what's next. If I can only get this to stop sharing, let me end this.
Thank you so much. That was perfect. We're going to move quickly to questions. And the first question, I believe, is about the need for testing in vaccinated asymptomatic individuals. Brad, do you want to take that?
Yeah, well, there's the need and the desire, and they're two different things. I think if you think about it from a personal health risk situation, you could argue that you're not getting the maximum utility out of doing that because, you know, you're probably going to be fine. But there is some advantage. We're seeing breakthrough cases, usually very rare that you'd have serious complications from those. So it is kind of important to know what's going on, but also we don't still have the evidence yet that you cannot transmit the virus. If you're fully vaccinated, you get a breakthrough case. I said that all along. We didn't have the evidence. We still don't have the evidence. I thought Israel would have published the evidence two months ago. It still is missing. We've seen high titers or high quantities of virus on people that have gotten to be a breakthrough case. We don't have information, though, about how long they remain viremic, but it's still theoretically possible. Therefore, knowing who is infected, whether you're vaccinated or not, is going to be an important element of trying to prevent transmission.
Thank you. There's a question about the constant section of the RNA and why that is not looked at for vaccines.
Yeah, slightly outside my wheelhouse. They're certainly looking at they're looking at regions that tend to stimulate the immune response, and I don't see why they shouldn't. I mean, some of the vaccines are much more cover much more of the virus than others. I don't know, Brad, is this your wheelhouse more than mine?
Not exactly, other than the fact that I know from the standpoint of serologic assessment, looking at some of the other some of the other antibodies that are formed, not just off the spike protein, have been a sort of deployed now for diagnostics at least. It is complicated. I think looking at some other targets is important, but apparently the spike protein was, I wouldn't say the easiest thing to do, but we know if you can block, attachment, that's going to have a maximum effect. But I'd leave it to some of our immunologists that could answer that question a little bit more competently. Not an easy thing to do. And the other thing that we still have only scratched the surface on, we really don't have a good understanding of the cell-mediated immunity component of what's going on with protection from the vaccines. We know it's important. It's not all humoral immunity. It's not all neutralizing antibody response, but we don't yet know what the other factors are that really need to be kind of teased out.
All right, great. There's a comment. Dr. Weimer, you left us hanging. Do you know what the next variants are going to be?
Not exactly. I mean, if I knew that, I would certainly tell you that. I would be telling everyone. I think it's a bit of a toss-up between 4 or 5 of them, actually. There's some really good websites out there that are publishing their data daily on which variants are circulating globally. And I showed you one. Richard showed you another one. If you want that, I'd go look at those websites and figure that out. The problem in the US is we don't necessarily publish that information in a timely way. And so it's very difficult to know what's going to happen locally. It's easier for me to figure out what's happening in the UK than it is for what's happening in the US, unfortunately.
Yeah, if I can jump on that. Yeah, Public Health England, it's an example of socialized health, but they have got a fantastic infrastructure. And if you're interested in this, you Google Public Health England, go to their website, and they publish almost weekly reams of data on variants and what's circulating, secondary attack rates, whether it's associated with travel or not travel. I mean, and the infectivity, I mean, it is remarkable what is available there, and it is remarkable what is not available in the US.
Yeah, it certainly helps. It certainly helps, Richard, if you have a health system rather than millions of systems. But no, I'm not taking anything away from them. You're absolutely right. It's an incredible treasure trove. Yeah.
Yeah. The other thing I would say, a lesson, getting back to the question to Bart, can we predict the next change? Now, the lesson from plant pathology is that bet on the side of evolution, right? Yes. We cannot predict the stochastic effects of all these mutations. And given the effective population size we have, the virus will explore just about every combination of amino acids that it still is viable with. And some things may last, the vaccines may last for a couple of years, they might last a couple of months, but ultimately there will be strains that overcome the vaccines, almost certainly.
Excellent. Okay, so this gets to Ryan's question. Are there broader efforts to align and advocate for data infrastructure pipelines from locality, state, national level? We know that the system is broken, but what's in play to fix it?
Well, there's quite a big effort going on at California level. Then the CDC through spheres is getting something together. It isn't really the problem again. It isn't one effort. There's maybe 2, 3, 4 efforts, and they aren't necessarily talking to each other. They're not necessarily fast. And the reason that CDPH is putting something together is because the national effort's too slow. One of the other problems we have with a national effort is you go to the lowest common denominator in terms of state restriction on sharing information. Right. And therefore the resolution you get and the type of information you get in a national effort is severely constrained to the point where it's almost uninformative.
So just repeat that, Richard.
So
states are
go ahead.
Unable to share. Say that one more time. Some
states are extremely restrictive on the type of information you can share on a sample. I mean, they won't even share the zip code. You can only look at the last 2 or 3 digits of a zip code and things like that. And if it becomes a national effort, the CDC will go with the lowest common denominator, right? The most protective. And so states that are a little bit more liberal are constrained by the paranoia of the most conservative state.
Okay, great. So now we have a question. Do we know how long, once infected, an asymptomatic or pre-symptomatic person can be infectious and spread? So back to the point about asymptomatic testing, that person, how long are they spreading?
Yeah.
Right. Well, one of the reasons that SARS-CoV-2 is so effective compared with MERS and SARS-1 is it's infectious before they're really symptomatic. And so the viral load ramps up quite quickly, 2 to 3 days after exposure. And they may not become symptomatic until 4 to 5 days. Now the viral load will continue to climb up to about 5 to 6 days and then usually declines. There's quite a good paper that came out of MIT Singapore whereby you can look at vaccinated individuals and the purging of the virus seems to be faster with vaccinated individuals than unvaccinated individuals. So there is that data.
Yeah, much faster. And that may be, so your time where you are at risk of transmitting with the vaccinated may be much shorter, even though the maximum amount of virus you're secreting may be equivalent, at least for a day or so. But we don't have the viral dynamics really nailed down as well as we should, though, in a fairly large cohort of folks where you're doing daily or even multiple times per day sampling, which would be very helpful to know in terms of the vaccinated effects.
So let me just put a headline on what you two just said, that in a vaccinated individual, the body purges more quickly and you're going to get less ill. So it is both.
Yeah, not less ill. No, not necessarily. I mean, you're just less infective than
less infective.
Yeah.
No, if you're right. I mean, if you're vaccinated, your chances of being hospitalized and the chance of dying are considerably less with vaccinated individuals.
Yes. Yeah. So the vaccination, number one, shortens your time frame of your infectivity.
Right.
2,
and yeah, saves you from dying and from being
and
hospitalized.
saves you from dying and being hospitalized. So if you're doing it for yourself and you're doing it for your ability to spread,
Yep.
but you're still, when you're vaccinated, you still should wear a mask, and you still ideally should be tested when and you can.
Yep.
This has been incredible. I can't tell you how many wonderful kudos you have in here. Excellent presentation, posted, etc. So Ana Lucia will be posting this. It will be external. Please share it, the link. We are so appreciative of all the commitment on the part of the speakers and so many, many others. We will be doing another one of these in 2 weeks, and we are going to try to do one shortly on vaccines. We think that would be a good topic. I am thanking you. It has been a whirlwind since April of 20. Unfortunately, we're still in that whirlwind, and we are all so appreciative. I wish all of you could see so many of the thank yous in the chat. Have a good weekend, and we will see you again in 2 weeks. Thank you so much.